Good morning, dear viewers. Today, I wish to talk on a very important topic of genetics and plant breeding. People used to get confused once handling an important trait for developing a variety. You see, this is the term linkage and Pleiotropy. These two terms are being confused while handling the important characters. So if you see about linkage, everybody know that linkage is the case that once many genes are located on the same chromosome and they are transferred from one generation to the next together. This is the case of linkage, and the genes which are transmitted from one generation to another generation together, they are called linked genes. So this is the case of linkage, and if you see the why it is happening, this is happening because as you know that characters are governed by the genes, and genes are located on the chromosomes, and the genes are transferred from one generation to next generation based upon the transmission of the chromosome during meiosis. So that is why you are getting linkage. So this is the one case, and if you take one example, any hypothetical example, say for in case of rice, we are seeking that the rice grains would have high zinc. If you are putting the gene responsible for high zinc, and then you are getting the result that high zinc, the variety is becoming low yielder. So high zinc, low yielder, whether this high zinc is the genes controlling high zinc is linked with the characters genes for poor yield, high zinc and poor yield. So this may be the case of linkage. Another is pleiotropy. Pleiotropy is the case where a single gene is controlling more than one character. So here all of us get confused whether the case of this high zinc and low yield. This is a hypothetical example I'm putting. A lot of examples are there. So whether this case is because of the pleiotropy. Pleiotropy is the case, as I said, that a single gene is controlling more than one character. So whether a single gene is controlling the high zinc as well as the low yield potential, or these two characters are governed by the linkage. So if you wish to try to understand, then you have to make the crosses. Of course, you are going to get the crosses between the high zinc and low yield variety, and with the another parent as a high yielder variety having no high zinc, low zinc content. If you are crossing these two, you are getting F1. Then you grow the larger population of segregating generation, particularly the F2 generation. Around 10,000 plants if you are growing, and if you are getting even a single gene, single plant, then you will be, if you are getting single plant, then you will be finding that, of course, you are the case was not a pleiotropy, the case was the your linkage, because it has broken. Linkage can be broken by allowing more and more combination, recombination, which is happening because of the crossing over and translocation during the meiosis. So in F2 generation, that is why you are in F1 you are not getting segregants. So in F2 generation and onward, you are getting segregants because of this recombination and translocation, because of the recombination and crossing over. So if you have allowed, and out of around 10,000 plant, if you have got a single plant even, then you can say that the case was, and the case is because of the linkage, and you have broken it. So linkage means these two traits, which is responsible for high zinc and low yield, they were located together on same chromosome, and they're being transferred from one generation to next generation together. So that is why you are not getting the high zinc and high yield. And because of the recombination out of crossing over and translocation, you have got in F2 generation a single plant or more plant. If you are getting single plant out of 10,000 plants, so it was the case of near about tightly linked genes. Although the tightly linked genes and pleiotropy both are very similar to each other, because by growing even larger population, you are not able to get a single plant. Generally, they are the case of tight linkage. Tight linkage means they are very nearby. The even in crossing over and translocation also, you are not going to get the desired recombinants. So this is the case. How to handle? If it is linkage, there is a hope that you can have the high zinc and high yield. But if it is the case of pleiotropy, you are becoming hopeless. So nowadays we are thinking that we should not be hopeless with this also, if it is the case of pleiotropy, because in case of pleiotropy also, the gene which is governing two traits, one good and one bad trait. So if you are putting in different backgrounds means in different genotypes, every genotype is different, genotype is having different genes, different types of combinations, different types of genes, different types of characters. So if you are putting, by crossing, if you are transferring these genes, these pleiotropy gene into different genotypes, then you are seeing the expression of that gene wherever you are getting the good combination, that high zinc and high yield. You should try to have that combination to develop a new outstanding variety with high zinc and high yield. Why in different background this pleiotropy is broken? It is not broken because in different background, in some genotypes are there which are having modified genes, or other combinations of genes which are allowing the high zinc but suppressing the low yielding ability and giving the high yielding characters. So that is why we are able. So we should not be hopeless with that oh, we have got pleiotropy, it is not the case of linkage, we cannot break the linkage. No. We should not be hopeless. We may get if we try with different genotypes combinations, and then if we try to see that, we can get it. So this was an important topic. I thought that I should discuss with all of you. Though I'm in Pune nowadays, in our holy vacation with our family. My son is living in Pune. So my daughter has come from Mumbai, so we came from Varanasi. It was a special days for India, celebrating holy festival. So we were together here. So yesterday was the Holi, we celebrated it, and I got little time today. I thought I should share this important topic with all of you. If you liked the talk, and if you have found this talk different from others, so please put your finger on like button, and if you have not subscribed the channel, please subscribe it. Thank you very much. All the best.